照片不错。
为什么是用它？
照片呢？
晚上抄来。哎。那么，
哎，你紧张吗？
不知道，突然有点不知所措。
我看到最这个位置能不能拍到？挺好。
哎。
Okay for you. No, that's fine.
Oh, wow. You can do that.
Did you take that yourself?
No, I just find it somewhere.
It's very nice.
Yeah. I don't know who.
I didn't see it.
Yeah, right. Oscillation.
Is he doing well?
Very well. He has something that's not clear, although it has to be like where I'm going to sit on the outside because I need to go revisit my unit as soon as we wrap up this skedaddle. Yeah, so they have this cool new technology for catching single rounds line fish. And they're full atlas. See what fish what's my 2 or 3 months.
I say we're doing 80% something.
That was it.
Yeah. Yeah, we record 80,000 simultaneously.
I just read his research report. He was similar. I think he's more interested in social traits.
So on this floor,
Why is it a PhD?
So they're not here, you should
In any case, he has every expectation that sell and not sell and so awesome. But
It's good that you got that.
it's kind of weird for me.
Fellowship and stuff.
He was my big thing. And all the advice that should be given.
I had no education.
So I just
I mean, 2 years ago.
I used
Yeah. Do they give more than 1 year? Because they give many each year. Several.
Yeah.
We'll put something happy about
Really divergent.
He's a buddy.
There's a student in my life and her husband. And he came from Scotland. I mean, his family, he was raised in California. But he was very, and for his thesis defense, he wore Scottish.
Flattered.
Thesis advisor. So, should apologize. I know we're going to have weird ethnic clothing.
The grad students are probably. No one in my tradition. Mike Wolf, Jeff Haskell. They'd rather do
How visible are you to the grad students? Are you teaching them classes?
I've taught in some random stuff, but I haven't taught. But I worked in that.
Yeah.
I introduced myself as Susan Thatcher, Jerry Stormo, Tim Shale, and a few other people. So hopefully some implications.
I just want to you're talking.
Yeah, well, she was a student in upstate, first try. So she's doing rotations now. So I have. I want to. My
Negotiations with Charlotte. So there are some
here.
So I guess for me, on my side, one more
Which I just got to be illegal. You play the hero that works no dice. HSG.
Is there a problem with getting that close? They're helping.
She's two.
I still have to do that, right? But then I only pick out two anymore and then I think so I just
Just one. No. Yeah. I don't know.
Yeah.
He was in a show. They're doing it this week. 4 o'clock tonight.
Are you serious?
We're still
One of the reasons we couldn't have a better tonight.
I can put it in the office next week. Well, that's a pretty good lead. Yeah, so she gets back in July.
And they're changing level.
Yeah, her Friday she happens for her jokes. Friday.
how are you?
By decree. Because we read out these. It's been back.
Oh, is it? This is
So he just walked in and spotted a lot of wildlife right in this moment. Oh, that's interesting. It's just momentous celebration.
I'm not sure exactly what this says, but
that was, I believe, aim 7 of his thesis was actually design and build.
Is this the first one? It's not the first one.
No, the observer was landed a long time ago. Yeah. Yes.
so this are all yeah, no, when I was sick. No, it's not.
I never walked in on the before I got sick. So I'm a little bit but it sounds like I'm actually working on a lot of the
We're living in a directory. Division.
It's just like this. And all the wood stuff.
There.
Roof on it
Has all of that.
really so they put bigger holes that satisfy all the water and the rest. And then they put it all wood and it's flooring. About a month and a half. Soon I was going to only take it. Thanks for asking. It's a big thing. Yeah, exactly. Shall I get things started? I wanted to kick off the festivities here briefly. You'll also hear from Paul Taghert as well. First, thanks to everyone for coming. This is a really, I think, momentous occasion because it marks the closure of a wonderful adventure of which Xitong has been the lead star. It's been a great pleasure for me working together with Xitong and Paul on this project. One of the things that I have enjoyed most about it was that it finally gave me a way to start learning about this beautiful organism, the fly, and all of the amazing things that you can do with all of the terrific genetic tricks that you can play in flies. I also actually have to confess a little bit of skepticism when the collaboration started, and that was simply that they came to involve me because of the use of light sheet microscopy. And one of the main reasons we developed light sheet microscopy was because it's really fast. And I thought, why on earth would you use a fast technique to study a slow process like a circadian rhythm? And I think one of the many things that I credit Xitong with was the gentle education, maybe you could say having my own words served to me on a platter over and over again, about how useful he has found this technique unique and how much he's been able to get out of it for the processes he's studied. He's found many fantastic ways to leverage the advantages of light sheet microscopy, even for slow systems. He's done some things that you couldn't do without the speed of light sheet microscopy. And he's really demonstrated, I think, ways in which his system has been amenable to analysis that have escaped some of the challenges of using it to tackle dense collections of neurons in mammals, which is what we use it most for. So I've really been educated by Xitong over the course of this project in many ways, and I'm immensely grateful to him for that. It's just been a terrific pleasure working with him and Paul, and I'm excited to hear his presentation today. Say a few words.
Yep. Thank you. Welcome also. Keep my remarks brief. I just want to congratulate Xitong. The academic calendar starts when you're young and enthusiastic, and then all of a sudden you wake up and it's time to defend your thesis, and that's today. So congratulations to you. It's been a great run. I want to thank Tim for being collegial, insightful, energetic, spontaneous, and always helpful. That's 4. Brief background: Xitong graduated from Peking University in 2012. He first came here as a summer intern in a program that Yi Rao started. Yi Rao was a professor in this department, and there were several people that came through. 3 of them are or are doing PhD studies here in neuroscience. Wei Kang and Jing Feng, you may remember, who's now, I believe, in Seattle. Both of these other students have been students with Larry Snyder. So Xitong stayed closer to the nest, and he's been conducting, as Tim mentioned, really pioneering work that he's going to describe for you today. I'll just say, I'm a little bit biased, but I think it's terrifically good, and it's really in the field of Drosophila circadian neurophysiology, redefined the terms of the playing field. To put it simply, previously everybody was interested in questions of how different neurons synchronize, and now Xitong has got everybody in the field to think about just the opposite. So it's hard to turn a field around on its end, but Xitong has done that, and hopefully in his remarks he will explain a little bit about what that means. I did want to mention that Xitong is terrific. Did I mention that? and I could go on, but the other thing you should know about Xitong, if you don't already, is he's extremely modest. So I would just embarrass him if I extended those remarks. Let me say one other thing. Circadian biology is all about anticipation. You may hear something about this. And in anticipation of a successful resolution to these deliberations this afternoon, we're going to have a small celebration. It's going to take place right across the hall, and it's going to start at Zeitgeber time 10. Tom? Okay. Yeah.
Hello, can everyone hear me? Okay. Thank Tim and Paul for a very nice introduction. Sorry. Yeah. Okay. So in general, I'm interested in how genes control behavior by regulating the functions of the neural circuit. So to study this question, I chose a relatively simple system, the circadian clock in fruit fly Drosophila, because in this system we have well-defined behavior, and we already know a lot about the underlying genes and the neurons. So in this system, we can really ask some questions about the links from genes to neurons, and the links from neurons to behavior. So today my talk will have 2 parts. So the first part is how the circadian genes regulate the physiology of circadian neuron circuit. And then the second part is then how the circadian neurons regulate the circadian rhythms in behavior. So let's look at the behavior first. So if you put the flies under 12 hours light and 12 hours dark light cycle, the locomotor activity of the flies will show this 2 peak pattern every day. This activity are measured by putting these flies in a tiny tube with an infrared beam detector. So each bar here represents how many times these tiny flies running back and forth and crossing this infrared beam within 30 minutes. And this this white bar the activity during the daytime, and the black bar the activity during nighttime. So every day you can see the flash will shows activity peak, allow the transient from night to day and from day to night. But these two activity peaks are not just response to environmental light on or light off, because the activity actually increased before the environmental LiDAR switch. So this is so-called anticipation. So the flies can anticipate the timing of environmental LiDAR switch and change their behavior before that. Since the flies know when the light will turn on or turn off, if you don't turn on the light at a constant darkness condition, the fly can still show this two peak pattern about the time when it's supposed to be morning and evening. So the fly know when it's morning, when it's evening, without seeing the sunrise and sunset. They keep the time by an internal clock. So to study the biological basis of this clock, about 50 years ago, people began to look for mutant flies, which lose the ability to anticipate the day-night changes. So in these flies, they can still show about the same amount of locomotor activity as the wild type, and they show response in their activity after light on and light off, but they just don't have this anticipatory behavior. By studying these mutants, people found a list of genes. Now we call this circadian clock gene. So we have two transcription factors which can activate the expression of a list of genes, including two they form so-called PER and TIM. These two proteins can form a dimer, and then they go back to nuclear to inhibit the activity of these two transcription factors. By doing so, they can shut down their own expression, and then over time, the two proteins will degrade in the cells, then the inhibition was gone, so this transcription factor then can activate the expression of these two proteins again. And then they go back to nuclear to inhibit themselves, and then they disappear. So this cycle takes about 24 hours, which then can make the expression level of these two proteins and also other genes under regulation of these two transcription factors will show circadian rhythm on their expression level. For example, this PER protein in the individual cells, it will go down during the day and goes up at night. So this specific cells then can know the time by just looking at this expression level of this protein. When the expression level is high, meaning it's the morning. When the expression level is low, meaning that it's evening. So by finding this molecular machinery for circadian clock, that these three gentlemen got Nobel Prize last year. Since we know on the molecular level how the clock works, so the major question of my thesis is to ask how then the circadian clock gene controls the circadian behavior. So people begin to study this question by just looking at where this gene is expressed in the brain. So here is a whole brain immunostaining against this PER protein. So we found that people found that out of about 100,000 neurons in a fly's brain, there are only a very small number of neurons express this clock. It's about like 150 neurons. So we call these neurons clock neurons. These neurons are required for the flies to have rhythms in their behavior. We also call them circadian pacemakers. So this clock neuron, they cluster into several anatomical distinct subgroups. Among these different groups, the molecular clock are actually very similar. As shown here in the PER protein expression level, it all goes down during the day and goes up at night. But in terms of controlling behavior, different groups of neurons might have different functions. So people found that these orange cells, we call them M cells, because they are specifically control the morning peak of the behavior. While these blue cells, they are specifically control the evening peak of behavior. So we call them E cells. And these two cells, in their functions in controlling behavior, can be quite independent because we can selectively kill either these two cells, and then we can selectively manipulate their behavior. For example, if we kill the morning cells, the flies will just lose their morning activity peak, but they still have an evening one. So the first question of my thesis is to ask how these different neurons with the same molecular clock can control distinct behavior phase at different times of day. So I try to answer this question by measure the daily pattern of this neuron's activity by in vivo calcium imaging with a light sheet microscope called OCPI in the living flies for 24 hours. So this OCPI, they use the light sheet to do the optical sectioning. So the light sheet only illuminates the tissue on the focal plane, which they can avoid the noise from autofocus tissue. And the light sheet also can illuminate the entire focal plane, so the camera here can, of the microscope can take the entire image at once, which then can be much faster and with higher photo efficiency than the laser scanning microscope, like 2 photon, which in order to take a 2D image, they need to scan across individual pixels. So use this light sheet, we then can scan the microscope together with the light sheet across the fry's brain. And we can do this several times a second. We can image the entire brain several times a second. So with a very small amount of light, which allow us to image from the living fries for 24 hours without causing too much photo bleaching and phototoxicity, and without disrupt the normal function of circadian clock, because the clock itself is sensitive to light. So we use intracellular calcium as a processor for neural activity. We found, which indicates by this GCAM sensor expressed selectively in these circadian neurons. We found that different groups of circadian neurons, which can be identified by their anatomical position, that shows a calcium activity pattern over 24 hours, and different group of neurons shows different calcium activity pattern. And then you may ask whether this pattern we found here really reflects a normal neural physiology or just an artifact, like this poor fries is just dying under the microscope. So we tested this by doing the same experiment at different times of day. So let's focus on these blue cells first. So each trace here are the calcium signal average from about 6 blue cells in one animal. And different traces are recording that in different animal. And the same 24 hours recording start at different times of day. The background indicates 2-day night cycle. You can see no matter when the recording starts, this group of cells, they always peak about the same time, before the night. So this calcium activity we measure here, they really change according to the circadian time, instead of like how long the fries is under the microscope. So we just average this calcium signal as a function of circadian time for different group of cells, which indicates by different color. We found that each group of cells, they all shows a calcium peak over 24 hours, but different group of cells peak at different times of day. Importantly, we have these morning cells, they control morning behavioral peak. It shows a calcium peak in the morning. While these blue cells, they control an evening behavioral peak. It shows a calcium peak in the evening. So different group of cells, they control different behavioral phases by being active at different times of day. And then we found this circadian rhythm in this neuron's activity are generated by the molecular clock within these cells. In the clock mutant fries, all these neurons lose their circadian rhythm in activity. So we then ask, how do the clock regulate this neuron's activity? So let's first take a closer look to this daily pattern of this neural activity. If we zoom in into specific time points with a high frequency imaging, here shows you one evening cells. At a daily peak time, these cells will show this high frequency calcium fluctuations. While at a daily trough time of their calcium rhythm, these cells will just silence. And because we can check the same neurons over 24 hours, in addition to this high frequency calcium activity, we also found a significant amount of change on the baseline level of the calcium between these different time points. And previous study, mostly done in mammalian circadian pacemaker neurons, shows several different ion channels was regulated by the circadian clock. So the circadian clock can generate circadian rhythm in the expression level of different ion channels. So some channels have higher expression during the day, some channels have higher expression during the night. So different combination of ion channels, they can switch the neural stability between day and night. So in the chapter of my thesis, I don't have enough time to talk about today. We suggest that a voltage-gated T-type calcium channel is important for this fast calcium activity, which suggests that this calcium fluctuation may be caused by the extracellular influx, which might be triggered by neural firing. While we also found this baseline change of calcium may be regulated by an IP3 receptor, which is a calcium channel on the ER memory, get the internal stock of calcium. So by this different pathway, we think the circadian clock then can regulate the neural activity by a cell autonomous way. However, in different groups of neurons, the daily pattern of the molecular clock is almost the same. So the question becomes how the same clock can generate different activity patterns in different neurons. And we think, although this rhythm of activity in these cells is generated by a cell autonomous mechanism, but their phase, the timing of the peak may be determined by some intercellular signaling. So we found one neuropeptide called pigment dispersion factor is important for this. So this PDF is released by morning cells and received by other neurons, including the evening cells. In the PDF knockout fries, they show quite abnormal circadian behavior. So we think PDF is an important output signal from the clock. And then we found in the PDF receptor mutant, the daily neural activity pattern of this circuit also become abnormal. They actually become more like their molecular clock. They look more synchronized. To have a closer look, I just plot the peak time of each group of cells on this 24 hours clock. In the wild type showed here, each dot here represents the peak time of one group of cells in one animal. And the arrow then point to the mean phases of each group of cells. In the wild type, different group cells peak at different time. Well, in the PDF receptor mutant, we found at least these three group cells, they become peak together in the morning, which is supposed to peak in the afternoon and at midnight. So we think PDF is important for this circuit to show this different phases activity pattern. So how? We hypothesize that the same molecular clock will generate a spontaneous activity peak, both in M cells and E cells in the morning. But when M cells become active, they will release this peptide PDF to inhibit the activity of the evening cells. And this inhibition can last for a long time, which can delay the activity peak in the evening cells by about 10 hours to make the E cells peak in the evening. So to test this idea, I tried to add some synthetic peptide to the knockout fries. So in this vehicle control, you can see in the PDF knockout, the purple cells and the blue cells peak together with the morning cells in the morning. But because PDF is released by the morning cells, so we first add PDF at the peak time of the morning cells to the brain, and we found by doing so, we can delay the activity peak of these blue cells and the purple cells by about 4 hours. And then if we add a second dose of PDF 4 hours later, we can further delay these two groups of cells. And we argue the effect here is to delay the activity peak instead of induce a new peak, because if we only add the PDF at a second time point after the peak of these two group cells, we will not able to induce a new peak. So in order to delay the activity phase, we need to add this drug before the peak rise. So together with other genetic manipulation, we suggest that the M cells use PDF to delay the activity peak in the E cells and in the DN3. So then we ask what are the phase of this DN1 and these red cells are determined. So we found a second neuropeptide called short neuropeptide F. When we knock down this short SNPF only in this circadian neuron circuit, we found that this green cells activity become arrhythmic. That now don't show any peak anymore. So you can see the arrow here become very short. It's because the dot is distributed around the clock, suggesting that the activity of these green cells now is now changed according to the circadian time. And this SNPF is released by the morning cells and by a subset of evening cells. So we then ask which one is more important for this green cells activity. We test this by selectively knocked out SNPF only in 4 morning cells. We found that in this case, this green cell shows a small but significant peak, but they peak at a wrong time. In the wild type, they peak at the night, but in this genotype, they peak in the morning. So this result suggests SNPF from both M cells and E cells is important for these green cells to have a normal phase. But what's the mechanism for SNPF to do this? Based on this phenotype, we come up with a model. So when M cells become active in the morning, they will release SNPF to inhibit activity in the green cells. And later, when E cells peak in the evening, they will also release SNPF to inhibit activity in the green cells. So the green cells activity will be suppressed throughout the daytime, so then it shows a nighttime peak. But when both M cells and E cells lose their SNPF, then the green cells will continuously active throughout day and night. But when only M cells lose their morning time suppression, then the activity peak of the green cells will then expand to the morning. So in order to test this model, I test two predictions from this model. So the first prediction is that the SMPF should be able to suppress activity in these green cells. So if we want to test this by adding the synthetic peptide to the brain, we will further predict that if we add the peptide during the daytime, it will cause a weaker response than we add the peptide during the nighttime, because during the daytime, the activity of these cells has already been suppressed by the endogenous SMPF signal. So we test this idea by just two flies from two different time zones, one from the day, one from the night, and we put them together in the same dish, and while doing calcium imaging, we add this drug. And as we expect, we found only from the flies from the nighttime, the green cell shows a significant reduce in their calcium signal in response to its SMPF peptide. And the second prediction is that in this arrhythmic genotype, the calcium level should be continuously high throughout day and night, higher than the daytime level in the wild type. So previously, we used GCAM as an indicator for calcium change over time in the same cells, but it's difficult to use this GCAM to compare the absolute calcium concentration between different cells because the GCAM signal might be changed, depends on the expression level of the sensor. So we use a different sensor called chameleon, which uses the ratio between two fluorescents to indicate the intercellular calcium concentration, which then can be independent by the abundance of the sensor. So we use this sensor, and then we sampled two time points in these two genotypes, and we found indeed in this arrhythmic genotype, the calcium concentration is continuously high, higher than the daytime level in the wild type, which suggests that the day-night difference in the wild type is because the daytime activity has been suppressed by some endogenous signal, hence it's this SMPF signal. So together, we suggest two different kinds of interaction within this circuit. So the M cells use PDF to control the phase in E cells and the DN3, while M cells and E cells together use SMPF to control the phase in this DN1. And these two different neuropeptides, they function in a very similar mechanism. They suppress other cells' activity. And one interesting future direction for this study is asking about what the phase of these red cells will determine. But to summarize this first part, I think I showed you that this circadian clock, by forming this feedback loop generating the circadian rhythm on the expression, they then can generate a circadian rhythm on the spontaneous neural activity in these circadian pacemaker neurons. And then different groups of circadian neurons, by their interaction, then different group of cells will peak at different time. So then the circuit together can send output at different time to control different behavioral phases. So the second part of my talk is going to ask then how the output of these circadian neurons controls the behavior. So in order to control this circadian rhythm in the locomotor activity, we think the output of circadian neurons should regulate the neural activity in some downstream premotor circuit. So what will be the premotor circuit? A large unbiased screening has been done in Genelia using thousands of flight lines label different subgroups of different groups of neurons, and then they can activate those neurons by expressing a temperature-sensitive CYP1 channel. And then they can compare the behavior outcome with the expression pattern of neurons they activate. So here I show you two examples from their data set. So this driver labels some neuron project to a structure called ellipsoid body. And this driver labels some neuron project to some structure called mushroom body. And when they activate these two groups of different cells, they will cause very different behavior outcomes. When they activate these groups of neurons, the flies will just running around on the arena. While when they activate these neurons, the fly just stand there forever. So I started these two movies at the same time, but you can see this fly just never move. So by thousands of experiments like this, then they can generate a statistic map for brain area which is more likely to induce certain types of behavior. So here is the statistical map for brain area. When they activate them, it's more likely to increase locomotor activity. So from this map, we recognize this structure. They have the strongest p-value. This neuron is called ellipse body ring neurons. So this neuron looks like this. They have cell body on both sides, and they send the process, and they have dendrite here, and then they send the process, the axon to the center of the frog brain, and then form a very interesting ring-like axonal terminal on a neural field called ellipse body. Since activating this neuron can increase locomotor activity in the flies, we asked whether this neuron's activity have any daily pattern. So we measure, we use a driver to label most of the EB ring neurons. This is the driver I showed you activated can make the flies just running around. And we imaged it for 24 hours, both under LiDAR cycle and under constant darkness condition. In both conditions, it all shows a strong rhythms in the activity. Importantly, actually, they shows two peaks every day, a smaller peak in the morning and a larger peak in the evening. Very like the fly's locomotor activity pattern, both under LiDAR cycle and under constant darkness condition. And this EB neuron can be divided into several subgroups based on different layers of their ring terminals. So we use several selective drivers to target each of these subgroups, and we imaging from them. And basically, we found all of them shows very similar two-peak pattern every day, a morning peak and an evening peak, just like the fly's locomotor activity. However, this locomotor activity I showed you are measured separately in this tiny tube with an infrared beam detector. In order to do similar measurement at the same time of my calcium imaging, I just simply put this infrared beam detector into the imaging chamber I use. Although in this case, the head of the flies was immobilized, we still use the leg movement as a processor for the locomotor activity, which shows in these two examples here, the black bar are the locomotor activity pattern over 24 hours, and the blue trace actually is calcium signal from one of the EB ring neuron subgroup. I hope you can appreciate the correlation between this calcium signal in the EB ring neurons and the fly's locomotor behavior. But just based on this correlation, you may ask whether it's the ring neurons activity drive the behavior, or on the other way, is the behavior drive the ring neurons activity, like by sensory feedback, proprioception feedback, right? So we test this idea by a kind of extreme way. So we try to remove the sensory feedback from behavior by just remove the body of the flies. And surprisingly, we found that in this body, this brain, this EB ring neuron still shows quite normal two-peak patterns, while the circadian neuron also shows quite normal daily patterns, which suggests that these rhythms in this EB ring neuron activity are not driven by sensory feedback from the behavior. So we then ask whether it's driven by the clock. So in this clock mutant, as I showed you, this fly can still move and it shows response to light on and light off. They just don't have this anticipatory behavior. And in this genotype, the clock neuron's activity was arrhythmic. And we found this EB ring neuron's activity are also arrhythmic. Even though these flies every day, they will show an increase in their locomotor activity after the light on and decrease in activity after light off. We don't see these patterns in this EB ring neurons activity, which suggests that the activity of this ring neurons might only encode those movements which triggered by the internal drive like the clock. They don't encode those movements triggered by this unexpected environmental stimulus. So, and then we ask how do the clock regulate activity in these EB ring neurons? The clock are only running in these circadian neurons, which don't include these EB ring neurons. So we think the simplest model is that we have M cells peak in the morning, then they can activate the morning peak in the EB ring neurons. And then we have E cells peak in the evening, and can activate the second peak in the EB ring neurons. So we test this model by two different ways. So first we ask whether there is any functional connection from M cells and E cells to the EB ring neurons. So we test this by selectively activate the M cells. We express ATP-gated cation channel only in 4 M cells. And then we perfume ATP to the brain. The ATP can only activate these 4 M cells. By doing so, we found when we activate M cells, we saw a response in the EB ring neurons. Similarly, when we only activate 4 E cells by expressing the channel only in these cells, we found we also see a response in the EB ring neurons. So this result suggests they do have some connection from M and E cell to the EB ring neurons. But do this connection really functions in a daily manner to generate this many hours peak? So we tested it by a different way. So we tried to selectively shift the activity peak in the M cells a little bit. So if the morning peak in the EB ring neuron are directly controlled by the activity peak in the M cells, then only the morning peak in the EB ring neuron should be shifted in about the same amount of time. So we achieved this by expressing a kinase called Shaggy only in these M cells to speed up the molecular clock in these cells. By doing so, we can selectively advance activity phase in these morning cells by about 4 hours without changing the evening cells phase too much. And we found in this price, the morning peak in the EB ring neuron was selectively advanced by about 4 hours, but the evening peak in these ring neurons was unchanged. As a consequence, we found on the behavior level in this price, the morning peak of in the behavior also selectively advanced, but not the evening peak. In parallel, when we do the same thing to the E cells to speed up the molecular clock in the E cells, we can selectively advance the calcium activity peak in the E cells by about maybe 3 hours, and we see the similar advance in the evening peak in the EB ring neurons, but not in the morning peak. So this result together shows that the morning peak in the EB ring neuron are controlled directly by the morning peak in the morning cells, while the evening peak in the EB ring neuron are controlled by the evening peak in the evening cells. And these two controls can be quite independent. But however, the connection from M cells and E cells might not be monogenetic, because actually we don't see an anatomical connection between these cells. So we think they might be mediated by some interneurons. So we look throughout the brain and find some interesting candidate new interneurons. So there are 2 pairs of dopaminergic neurons called PPM3-EB. So this PPM3, they also project through this green-like structure. Importantly, they have this 2 peak pattern every day, just like the activity pattern in the EB ring neurons. And based on anatomy, we hypothesize that maybe the output of M cells and E cells may converge into these 2 PPM3 neurons. And then these 2 PPM3 neurons then can modulate the activity of about 60 EB ring neurons. So to test this model, we try to block the output from these 2 PPM3 neurons. So if this diagram is true, then it should block the circadian regulation from clock neurons to these EB ring neurons. So we do this by express a tetanus toxin only in these 2 PPM3 neurons to block the neurotransmission of these cells. And we found first in this price, the locomotor activity become largely arrhythmic under constant darkness condition. It's quite surprising to me because we only manipulate the activity of 2 neurons. We were never able to get such a strong behavioral deficit by manipulating 2 circadian neurons, which suggests that this 2 PPM3 neuron is an important convergence point of the output of the circadian neurons. And importantly, we found in this slide, the daily activity pattern of EB ring neurons was disrupted, but the daily pattern of the circadian neuron is still quite normal, which supports the idea that these PPM3 neurons mediate the regulation from circadian neurons to these EB ring neurons. So to summarize this part, we think the molecular clock will generate a spontaneous activity peak in the circadian neurons, and because M cells inhibit the E cells, then 1 peak in the morning, 1 peak in the evening. And each of them can activate the PPM3 neurons to generate this 2 peak pattern in this PPM3 neurons. And the PPM3 neuron can free forward this 2 peak pattern in this premotor EB ring neurons to generate the 2 peak pattern in the fresh locomotor activity. But just like in the humans, there's many different physiological functions and behavior have circadian rhythms, like the body temperature, blood pressure, different kind of hormone release. They all show circadian rhythm. And we think the flies also need to do different things every day and do the right things at the right time. So in addition to this locomotor activity, they also show circadian rhythm in the sleep, feeding, and the mating behavior. So we will ask how these different behavior are regulated by the circadian clock. We think they might go through different downstream pathways. And to looking for these downstream neurons, we just try to look throughout the brain for neurons which show circadian rhythm in their activity. And then we can try to link their daily activity pattern with their different functions in controlling different behavior and physiology. So we're looking at some candidate system, like some neuromotor system, like dopaminergic neurons. So dopamine, in addition to regulating the locomotor activity, they also can regulate the sleep. They will suppress sleep, promote arousal. They also regulate like feeding and mating and learning and memory in the flies. So we're imaging from the dopaminergic neurons, which also cluster into several subgroups. And we found that most of them shows the circadian rhythm in activity with diverse phase. So some peak in the morning, some peak at the midday, some peak in the evening. In addition to dopamine, we also look at a different neuromodular system, the neuroendocrine cells, the peptinergic neurons. And we also found different groups of neurons that also show circadian rhythms with different patterns. Some peak in the morning, some peak in the evening, many of them peak around the midday, which can be responsible for the circadian rhythm in the release of different kinds of hormones. In addition to this candidate approach, we also did a systematic survey by doing whole brain pan-neural imaging. So we express the GCAM in all the neurons, and then through the cranial window, we use the light sheet microscope to image the entire brain for almost all the neurons, and then for 24 hours. And then I use an algorithm called JTK cycle to analyze this volumetric data. So vessel by vessel, I use this algorithm to detect the circadian rhythmicity of calcium signal. And then I can generate this statistic map for brain area which are more likely to show circadian rhythm in their calcium activity. So the brighter color indicates those areas might have stronger rhythms. And then I can color code those regions with their phase of their peak time. So we can see this phase map on 3D. So this bluish, purplish color indicates this brain area peak about the midnight, while the greenish color indicates those areas peak about the midday. You can see the phase distribution is quite diverse across the brain, but it's kind of stereotyped between different brains, which these three different flies was recorded from different times of day. And we also found this pattern was largely disappeared in the clock mutant flies, which suggests this pattern is really generated by the circadian clock. So in summary, we found the most brain volume that shows the activity peak around the nighttime, while a small proportion peak around the day. From those areas peak at the night, the strongest signal actually comes from this brain area highlighted by the dashed line. It's so-called antenna lobe in the flies, which is the first order olfactory center in the flies. And it is kind of a surprising coincidence with previous study showing that the flies olfactory sensitivity, it shows a day-night difference, that shows a peak about the midnight. And this pattern is controlled by a clock, so disappeared in a clock mutant. And then I want to just summarize some of my findings and mostly hypothesis and future direction in these slides. So we think different group of circadian neurons, they peak at different time. And their peak time, they can send output to different downstream circuits to control different behavior and physiology. So we use the power of fly genetic to selectively shift the activity peak in each of these cells. And then we're looking at the activity pattern in different downstream circuits from those data, we then can have some indication about which downstream neuron is controlled by which circadian neurons. And as I showed you before, we found the output of the morning cells will generate the morning peak in this PPM3 neuron, and then go through the EB ring neuron to generate the morning peak in the locomotor activity. In parallel, the evening cells output will generate the evening peak in the PPM3 neuron, which then goes through EB ring neuron to generate evening peak in the locomotor activity. In addition to this, the output of the evening cells might also activate an evening peak in a different group of dopamine neurons called PPL1. And this neuron has been shown in regulating sleep. They can suppress sleep and promote awake. And here shows you the daily sleep pattern of the flies. If we flip this curve, the dashed line indicates the daily pattern of wakefulness in the flies. And based on the daily pattern of this PPL1 wake-promoting neurons, it raises an interesting future direction to test whether this PPL1 is specifically to control, to wake up the flies in the evening. And similarly, in addition to activate different downstream neurons, we also found the output of evening cells might inhibit a different group of dopaminergic neurons. So instead of generate the evening peak, they will generate the morning peak pattern in this PAL dopaminergic neurons. And this PAL neuron has been shown in promoting mating behavior in the flies. So at least based on the superficial similarity between the daily pattern of these PAL neurons and the daily pattern of mating behavior in the flies, it also raises an important hypothesis, an interesting hypothesis that maybe this PAL neuron mediates the regulation from the evening cells to generate this daily trap in the mating behavior in the flies. We also have evidence showing that these red cells, they peak at a midday, they may generate some midday peak in several downstream neurons. And these purple cells and the green cells that peak at the midnight, they may work together to generate this midnight peak in the antenna lobe neuron, which then can contribute to the circadian rhythms in the olfactory sensitivity in the flies. And we have one more candidate downstream neurons. This neuron release insulin-like peptide, and they shows a morning peak in the activity, which likely to be regulated by output of the morning cells. This insulin neurons has also, just like in vertebrate, that they might also involve in regulating the feeding behavior. Since these two parts, their daily activity pattern is very similar to the feeding behavior pattern, it suggests that this insulin neuron might also mediate the circadian regulation to the feeding behavior. In addition to regulating feeding behavior, this insulin neuron also has been suggested to wake up, they can suppress sleep in the flies. And since we have these cells waking up the flies in the evening, and these cells peak in the morning, which might suggest that this insulin neuron might selectively wake up the flies in the morning. So looks like the flies have two different alarm clocks, which one wake up in the morning and the other one waking up in the evening. So together, I think my study began to come up with a few very interesting future directions about different output pathway downstream of the circadian neuron circuit, which can be tested in the future. So to summarize my entire thesis, I think we begin to show that the circadian clock gene, by forming this feedback loop, will generate a circadian rhythm in a spontaneous activity, first in circadian neurons, and then this rhythmic activity will spread out and then generate diverse pattern in neural activities throughout the entire brain. And then different neurons with different rhythms then can generate circadian rhythms in different physiological functions and the behavior. So with that, I will begin my acknowledgement. So in general, so first I would like to thank Paul and Tim and everyone in the lab. I also want to thank my thesis committee. First, Eric is one of the best teacher I ever have. I was been in a TA in his class, and later on I also attend one of his undergraduate class, the biological clock. And I really learned a lot from not only from what he want to teach, but also from how he teach them. Yeah. And then I want to thank Masa and Daniel, them as electrophysiologists that really pushed me very hard to think about the neural property of my cells, the cells I study. So in order to convince them that the circadian pacemaker cells, that they are really neurons. So I really, yeah, with the insight, I really think a lot about the cells I'm studying. And then I would like to thank also our collaborator, Mark Wu and his student. They share their insights in dopamine biology and sharing us with some reagents, especially for the second part of my thesis. I also want to thank Ori Schaeffer for he is always the first reader of my manuscript, and he really provides a lot of helpful comments. And also Eric Herzog is also always the first reader of our manuscript and provide a lot of comments. And I also like to thank people from all over the world sending us transgenic prize. And I would like to thank Dennis from Uchi who training me on different kind of microscope. I would like to thank the clock community in WashU, especially Matt. So before I joined the lab, Matt taught me, you should join this lab, then we can talk about circadian clock. So yeah, so actually we have dinner every week before he graduated a couple months ago. So we have a lot of discussion. And also Matt also take care of me very well when we went to different kind of circadian conference together. I also want to thank Jia. She teach me about like the JTK psychoanalysis and also share with me some prize. I also want to thank Christina, Jeff, and Krishna with very helpful discussions. And then I want to specifically talk about Paul. Sorry. So I think my thesis project is basically all his idea from the beginning. From the beginning, I mean far away from my joining the lab. So I heard that this project was in the K99 proposal of our former postdoc in the lab about 10 years ago. So like 10 years ago, Paul had begun to think about this project. And Paul initiated the collaboration with Tim, and then he was fully involved in every step of this project. And also I want to share a story about Paul, maybe he don't know, is our first interaction was like in the winter 7 years ago when I was contacting him, applying for working in his lab for a summer. And at that time, I'm really upset at that time. I have been sick for a long time, like for half an year. And sorry. I've been sick for half a year and almost got kicked out from my previous undergrad lab because I absent for a long time. And I applied for several prizes for summer internship and almost got rejected for everyone. And so I feel so lucky. Paul said, yes, you can come. And I really had a great time, great summer here. Thanks to Paul and thanks to people in the lab. They treat me so well, so they bring me back here for graduate school. And yeah, and I'm not the only one who benefit from this summer internship. So over 8 years, Paul have 9 students coming from my undergrad department. And so here is a small reunion at the year I joined this program. So some previous students come back to St. Louis. And after that, we also have Tian and Wei Kang visit the lab. So we all really appreciate you about this wonderful opportunity. And then I want to thank people in Paul Taghert's lab. So we are such a small lab. So in the past 5 years, most of the past 5 years, we only have Wei Hua and Jennifer. So I really appreciate them for taking care of me, helping with my experiment and helping with my life. And I would like to thank people in Tim Holly's lab. So Tim is the greatest scientist I ever met. And I'm not the only one saying so. I met many people when I interviewed for postdoc in many places, and they all say the same thing. He is the greatest scientist. So so my undergraduate mentor is the guy who kicked me out. He told me you can never become an expert in everything. I think in general that's true, but Tim is just an exception. He is an expert for everything. He can just become an expert for anything in a second. So I really I don't know how much I can learn from that, but I really benefit a lot from that, to have a mentor who knows almost everything. And then I would like to thank people from Tim Holly's lab, because I'm the only student in post lab, so I go to lab meeting in Tim's lab every week. So we have very helpful discussion there. And especially I want to thank Cody for building the new generation microscope, the light sheet microscope. I use that a lot, and every time I meet a problem, Cody is always very helpful, and he just solves the problem very quick. And then I want to thank Daewoo. He upgraded the software for the microscope, especially with some feature designed for my purpose. And then I want to thank Tara. She supervised me when I rotate in the lab, and since then he provided me a lot of generous advice. I also want to thank Jerry. Jerry is also very helpful. I remember several times I just emailed him with a question about midnight or 1 or 2 a.m. and he just responds immediately and solves the problem. Yeah, he's super. Yeah. And then I want to thank all my friends in St. Louis. I really have a lot of fun together. Especially I want to thank my roommate Qi Hao and Cheng Cheng. We really have a great time living in the same apartment. And then I want to thank my animal friends. We used to have up to 4 cats. So this is Toothless, this is Sophie, and this is Latte, and this huge cat his name is Cat. And we also have a bird, although her life is always in danger surrounded by cats. And we also have a chameleon. When he just came, he's just so small, and now he is huge. Yeah, so I really appreciate it. And I also want to thank my parents. They always support me and encourage me to try whatever I want. And so I left them far away from home for college and then for graduate school. So since then, we only met 1 or 2 a year, so I really miss them. So I hope I can graduate and go back to China. Yeah, and finally, I want to thank my girlfriend, Xueying, and we know each other in college, and I feel so lucky that we can come together to watch you for graduate school, and we really, thanks to her, I really had a great 5-year PhD. Thank you. And yeah, with that, I'm happy to take any questions.